Hello everyone. In this video, we're going to talk about molecular pathogenesis of malignant mesothelioma. Malignant mesothelioma is an uncommon and deadly malignancy that arises from the mesothelium that covers the body's serous cavities such as pleural, peritoneal, pericardial, and tunica vaginalis of the testis. There are three major histological subtypes of malignant mesothelioma, which are epithelioid, sarcomatoid, and biphasic types. Epithelioid is the most common subtypes and resemble normal epithelial cells, which form the lining of organs. Sarcomatoid subtypes consists of spindle-shaped cells and is associated with a more aggressive form of mesothelioma, while biphasic subtypes consist the mixture of both epithelioid and sarcomatous cells. Malignant mesothelioma is frequently associated with asbestos exposure that can be classified into two major groups, which are the amphiboles group that are sharp, needle-like fibers, and the serpentines, the group that are curly fibers. Asbestos fibers are inhaled into the body via nostrils and mouth. If the long and thin asbestos fibers manage to evade lung and reach the alveoli, macrophages will attempt to engulf the asbestos fibers and undergo phagocytosis. In some cases, macrophages fail to engulf the fibers due to their long needle-like structures. Fibers that manage to evade phagocytosis will translocated to the mesothelium. There are several possible mechanisms involved in how asbestos fibers cause malignant mesothelioma. Firstly, reactive oxygen species ROS that are generated by asbestos fibers lead to DNA damage and strand breaks of the cells. Macrophage, which phagocytoses asbestos fibers but is unable to digest them, can also produce abundant reactive oxygen species. Secondly, asbestos fibers are also engulfed by mesothelial cells. Tangling of asbestos fibers with chromosomes or mitotic spindles may result in chromosomal structural abnormalities and a neoploidy of mesothelial cells. Thirdly, asbestos fibers can absorb a variety of proteins and chemicals, which may result in the accumulation of hazardous molecules, including carcinogens. Lastly, asbestos-exposed mesothelial cells and macrophages release a variety of cytokines and growth factors, which induce inflammation and tumor promotion. Inflammation response leads to DNA damage and cause tumor development. As we know, malignant mesothelioma happened because of asbestos exposure. Just like mentioned just now, asbestos fibers are inhaled into the lungs when individuals are exposed to asbestos. So once in the lungs, these fibers can become embedded in the lung tissue. So macrophages, which is immune cells present in the lungs, will attempt to engulf and remove the asbestos fibers. However, this asbestos fibers can resist degradation, leading to persistent inflammation. Once asbestos fibers are inhaled and reach the lungs, immune cells, particularly macrophages, recognize them as foreign bodies. The presence of asbestos activates the immune system, initiating an inflammatory response as the body attempts to defend itself against the perceived threat. In response to asbestos exposure, immune cells release various signaling molecules known as inflammatory mediators, such as cytokines and chemokines. The example of cytokines is interleukin-1 and tumor necrosis factor alpha. Inflammatory mediators that I mentioned just now will attract immune cells to the site of asbestos deposition. This includes macrophages, neutrophils, and lymphocytes. These immune cells will release additional pro-inflammatory signals and create a microenvironment characterized by chronic inflammation. Asbestos exposure will induce the production of reactive oxygen species within cells. ROS are highly reactive molecules. Examples of ROS includes molecules like superoxide radicals, hydrogen peroxide, and hydroxyl radicals. The presence of an elevated level of ROS leads to a state known as oxidative stress. Oxidative stress occurs when there is an imbalance between the production of ROS and the ability of the body to neutralize these molecules. Oxidative stress can lead to the damage of cellular components, including lipids, proteins, and also DNA. Oxidative stress that caused by ROS and direct interaction with asbestos fibers will contribute to the DNA damage within mesothelial cells. DNA damage can manifest as single-strand breaks, double-strand breaks, and also the formation of DNA adducts. DNA damage compromises the integrity of the genetic code within cells. Unrepaired or misrepaired DNA lesions can lead to mutations and chromosomal aberrations. Inflammatory cytokines, particularly those released during chronic inflammation, can activate the signaling pathways within cells. Activation of pathways such as nuclear factor kappa B and activator protein 1 can promote cell survival, proliferation, and the release of additional inflammatory signals. DNA damage and the persistent inflammatory microenvironment causing the genetic and epigenetic alterations in mesothelial cells. Mutations can occur in critical genes involved in cell cycle regulation, DNA repair, and tumor suppression. For example, the tumor suppressor gene p53 is frequently mutated in malignant mesothelioma. Mutations in critical genes like p53 and CDKN2A can lead to the loss of normal cell cycle control. Dysregulation of the cell cycle allows for uncontrolled cell growth and proliferation, contributing to tumor development. Oncogene activation cascades play a significant role in the development and progression of mesothelioma. Oncogenes are mutated forms of proto-oncogenes, which are genes involved in normal cell growth. When oncogenes are activated, abnormal cell growth occurs, leading to the development of cancerous cells. Receptor tyrosine kinases (RTKs) are involved in mediating cell-to-cell communication and control a wide range of complex biological functions, such as cell growth. In malignant cells, RTKs are frequently activated, leading to the continuous activation of two pathways, the RAF-MEK extracellular signal-regulated kinase pathway and the phosphoinositide 3-kinase P13KAK-T pathway. These pathways are critical for the proliferation and survival of cells. The activation of mammalian target of rapamycin mTOR signaling contributes to the pathogenesis of many tumor types, which is also one of the P13KAK-T downstream pathways. In mesothelioma, rapamycin and mTOR C1 inhibitor is known to enhance cell death with cisplatin on malignant mesothelioma cell lines. In addition to these pathways, the signal transducer and activator of transcription, STAT1 signaling axis, has been shown to be activated in malignant mesothelioma cells. Although STAT1 is considered a tumor suppressor, it has also been shown to promote radioresistance and tumorigenesis. Cyclin-dependent kinase inhibitor 2-CDKN2A is a gene that provides instructions to make several proteins and is considered the most frequently inactivated tumor suppressor gene. The CDKN2A encodes for proteins P16INC4A and P14ARRF. The P16INC4 binds to CDK4 and CDK6, which are proteins that stimulate cell division, and block CDK4 and CDK6 from stimulating cell cycle progression. On the other hand, P14 protects P53, a tumor suppressor that regulates cell division. Thus, the homozygous deletion of CDKN2AARF indicates the inactivation of two major tumor-suppressing pathways in the cell. In simpler terms, exposure to asbestos causes the inactivation of CDKN2A, which then inactivates the tumor suppressor genes in our body, leading to uncontrolled cell division and eventually resulting in malignant mesothelioma. Next, consider neurofibromatosis type 2 inactivation. NF2 is a tumor suppressor gene that encodes the protein myelin, which acts as a tumor suppressor by inhibiting a specific protein in mesothelioma cells, in turn inhibiting the function of the protein in the modification of its target protein. Inactivation of NF2 leads to the loss of myelin's tumor suppressor function, allowing uncontrolled cell growth and division, which can contribute to the development of malignant mesothelioma.